Today we are going to discuss the embryology of angiosperm. As earlier we discussed, those two units belongs to medicinal plants. And the last class we discussed the embryology introduction and male gametangium and all anther structure and development of male gametophyte, male gametangium. That means microsporogenesis. Microsporangium structure, microsporogenesis, microspore pollens type, cypress pollens type, nemagnomina, ovulus stamen type. That all we have discussed. Now, today we will discuss the megasporogenesis, megasporangium, ovule, megasporangium or ovule. That we will discuss. Okay. Today we will discuss the megasporangium. Megasporangium, that is nothing but ovule. Nothing but the ovule that we will discuss. Okay, megasporangium or ovule. The structure, types, and functions. Ovule, that means megasporangium, structure, types, and functions. And megasporogenesis, how the megaspores are generating, types of the megasporogenesis. And actually in embryology, we have a lot of syllabus we have to complete as soon as the next week class. Microsporogenesis, male gamete, megasporogenesis, ovule, and fertilization, apomixis, parthenocarpy, and then what are there in the syllabus? Fertilization, megasporogenesis, microsporogenesis, parthenocarpy, apomixis, polyembryony, endosperm type, and role of embryology in taxonomy, horticulture, agriculture, and palynology, applications of palynology. That all the topics are there. Now we are discussing the structure, types of the ovule. You know the ovule, where it is? Where the ovule? You know the flower? In the flower, gynoecium, androecium, sepals, petals, pedicel. These are the general floral characters, floral parts. In this floral part, we already discussed essential organs and non-essential organs. Sepals and petals are non-essential organs. These are not useful in the fertilization, after fertilization, only for the attraction, only for the covering, only these are useful. And what are the essential organs? That are gynoecium and androecium. These two are essential organs, sexual organs. Androecium is nothing but the male reproductive organ. Gynoecium is nothing but the female reproductive organ. Gynoecium. Gynoecium again divided into three parts. What are those three parts? Ovary, style, stigma. Ovary, style, and stigma. These are the three parts. Ovary, it is a sac, in that it has the ovules. Ovary will become the fruit after fertilization. After the fertilization, ovary will become the fruit, ovule will become the seed. Megasporangium or ovule. In the ovule, ovaries are present. Based upon there are so many types of the 6 types of the ovaries types, ovules types are there. That also we will discuss. And structure of the ovule also we will discuss. How the structure of the ovule. You see the structure of the ovule, how it is. This ovule is a female gametangium. We also call it a female gametangium, also called as megasporangium. It having the megaspore ovule inside that megasporangium. Ovule also called as megasporangium. Anther also called as microsporangium. I already told you the anthers from the microsporangium reaching toward the stigma. Pollens reaching toward the stigma is called as pollination. And fusion of the pollen, that means male gamete with the female, is called as fertilization. That fertilization also one of the big essay question. What is the fertilization? Types of fertilization and the process of fertilization, steps in the fertilization. That also very important. And fertile embryo sac, embryo sac which is ready to fertile, also very, very important essay question. These 3-4 questions are there from this megasporangium. Write about the megasporangium structure and type, and write about the megasporogenesis types and fertilization types and process of the fertilization in angiosperms. These questions raised from this chapter. Okay. Now you can see the structure of the ovule, how it is. You see where in the ovary ovule is present. In the ovary, ovule is present. The ovule, it is usually arranged in the middle. It having the style and stigma, you know, the style and stigma will be removed after fertilization. For the pollination, it needs the stigma to reach the pollen toward the stigma. Then after the style in the tube, it will reach toward the egg apparatus, egg cell. That is called as fertilization. After fertilization, style and stigma will be removed and the ovary will become the food. In that female gametangium, that is embryo sac, will be remain. That will become the embryo sac after fertilization. The egg cell after fusion with the male gamete for pollen, that will become the seed. The seeds are there inside. That is came from the syngamy after the syngamy reaction, fusion of male and female gametes. That's why the seeds are diploid. Seeds are diploid. One N from male, one N from female. N plus N to N. Okay. There you can see here in this ovule structure, there is a stalk-like structure which is attached to the ovule is calling as funicle, which is attached. Funicle, it is attached to the ovule. Okay, that we're calling it as funicle. And you know, the hilum The competitive exams, so many competitive exams they ask. Hilum. Hilum is a attached point of the funicle with the ovule. Funicle with the ovule. Funicle with the megasporangium. Stalk with the megasporangium. Where there is attached point, that we call it as hilum. Whenever the hilum, even after touching ovule, it goes further and it touches toward the ovules up to the chalazalan. That extra growth of the funicle is calling it as raphe. Extra growth of the funicle is called as raphe. You might have seen in the beans all the pulses. The pulses seeds, red gram, green gram, black gram, whatever it is the seed, you might have seen the white patches like structure of the micropylar end. White micro structure, extended funicle, that we call it as raphe. That is present mostly in the most of the pulses. Red gram, green gram, moss gram. You might have seen white appendages structure. That is raphe. That is present in the pulses. Okay. And then especially in the ovule structure, what are the parts are there? The outermost part is integument. Is a integument layers, protecting layers. The protecting layer, integuments. Okay, that integument, based upon the integument, three types are there. Ategmic, that means no tegma, that means outer layer, protecting layer. Bitegmic, that means two layers of the tegma, outer and inner layer. And polytegmic, that means more numerous number of the tegmas, protecting layers. Three types are there. Usually in the Ranunculus, Bitamaceae, Olistamaceae, ategmic ovules are present. Okay. And then where in the ovule, there is a two ends are there. One is micropylar end, another one is the chalazalan end. Where the integuments are open up with the porous space. Outer and inner integument. These are open up. These are open up with the porous space. That open porous space of the integument is calling that micropylar and micropyle under dwaramu. From here, the male gamete will go toward inside toward the egg cell, toward the egg cell. That is micropylar end. The other end, where the integuments are end, that end is called as chalazal end. Chalazal end and micropylar end. You can see always the egg cell operators toward the micropylar end, and antipodal cells will be there at the chalazal end. Antipodal cells will be present in the chalazal end, and egg operators will be present at the micropylar end. So many complex examples. Egg operators will be present in which end? Chalazal end or micropylar end, at the middle, or in the integuments, where it is. They ask the question. Okay. Here you have to write the essay questions and short answer. Write about the structure of the ovule. Then you have to draw this diagram. You have to explain about this part. Funicle, hilum, raphe, integuments, chalazalan, micropylar end, embryo sac, and endosperm, embryo sac, nucellus, endosperms. This all we have to explain. Okay, these all are the parts of the ovule. These all are the parts of ovules. Okay, and then here you can see clearly in the flower how the structure is. I already drawn this. You see the ovule, ovary, style, and stigma. How it is. You see, if you cut any part of this, that will be look like this ovule. It having a lot of might be a lot of ovules. One single ovule or multi-ovular. That means single-seeded or multi-seeded. Single-seeded or multi-seeded. You can see. Or the seed coat we calling it as textron tegment. But here ovule coat we tegment. Inner integument and outer integuments. Okay. Inner and outer integuments. Where they is having the porous space, that is called as microphylla. And where the open up space, that is chalagial end. And in between the embryo sac and the integument, there is a nucellus. Nucellus is present. That's also a question. Nucellus types of the nucellus and importance. And endosperm, which is forming after fertilization in the embryo sac, that endosperm also is a question. Endosperm will be formed after double fertilization, that means triple fission. Triple fission. That means two polar nucleus fusing with the one male gamete. Three nucleus are fusing. Triple fission. This triple fission was discovered by Navasin. Who is that? Navasin. Two polar nucleus of the ovule and one male gamete. Three nucleus are fusing. That means secondary male gamete. That is called a triple fission. Triple fission first discovered in the Hyacinthus orientalis by Navasin. Okay, the first fusion is fusion of the male gamete with the egg cell apparatus at the chalagial end. Fusion of the 1 plus 1. 2n. Singami. Singami. Singam, not Singam. That Singami is the actual fertilization. And the second one is the triple fusion. These two commonly called as double fertilization. Double fertilization, it will take only in the angiosperms. Double fertilization will not take place in the gymnosperms. In the gymnosperms, only the fusion of male gamete with the female will be happen. Before the fusion of the male gamete with the female, whatever the ovule cells, female gametophytic cells that take the multiple divisions, those are applied endosperm will be formed. Applied endosperm will be formed. That is the difference between gymnosperm and angiosperm. Okay. And then we also can see the structure, what are the parts of the ovule. Okay. You see the structure, anatomical structure, how it is, how the ovule under the microscope. You can see the integuments. Here we can see the integuments. These integuments we can see. And the nucellus and the embryo sac. Female gamete, embryo sac. This all you can see clearly even under the microscope. That is the structure under the microscope. You can see the difference between gymnosperm ovule and angiosperm ovule. What are the difference? Can anybody say? Difference between gymnosperm ovule and angiosperm ovule. What it is? What is the difference? You see, main distinguishing feature is it having the what it is having protecting layer, outside layer. You can see this one, whereas the gymnosperm having only this directly ovule, naked ovules are present, naked seeds are present. Whereas here in the angiosperm, it having the ovary wall, extra ovary wall, ovary seeds are present. That's why these are angiosperms, these are gymnosperm. Naked seeds will be formed with the structure of the ovule. Naked seeds will be formed. Here you can see the nucellus in the same boat and the megasporophyte, megasporangium, egg cell is there. This megasporangium will take the meiotic and mitotic division and it form the 8 nuclei, 7-cell embryo sac. Normal type of the embryo sac is polygonum type. Polygonum type of the embryo sac is present in almost all the angiosperms, most of them. But some of the variations are there. Cyphorus type and another type, so many Adaxa, Pinia, Peperomia, Plumbago, Plumbagella, Fritillaria, these all are the types of the embryo sacs are there. That also we will discuss under the development of embryo. Under the development of embryo, we will discuss monosporous, bisporous, and tetrasporous type of the embryo sac. That also we will discuss. Here is extra sheath is there, ovule sheath is there in angiosperm. It is absent in the gymnosperm. That you should remember. Okay, difference between the gymnosperm and angiosperm. Here you can see, I already told you, it is an oval shaped structure. It originates from the placenta, lies inside the chamber of ovary. Inside the chamber of ovary. Okay, in the chamber of ovary, ovules are present. Either 3, 4, 5, 6, 7, whatever it is. Okay, each chamber may consist one or more number of the ovules. Okay, and the ovule is having the stalk, funicle. I already told you the funicle stalk. It attached to the ovule. Funicle, where it attached, that place we calling it as hilum. Okay, where the funicle it extends toward the base of the ovule, that is called as raphe. That is called as raphe. We already discussed this all. Okay, and then where is the annotation? What happened to this? Yeah. What happened? What happened to this? Here you can see easily the structure of the ovule, but having the most important structure, the structure of ovule, where we are discussing, we are seeing the diagram that you can see here. Here, the ovule structure, integments we discussed, and then you can see how many integument based upon the integument. Three types are there that we already discussed: ategmic, bitegmic, and polytegmic. You see, if there is no integument, that is ategmic. If the two integuments are unitegmic, only single integument, or two integuments, bitegmic. Okay. Here you can see where the integuments ended, that we call it as where the integument is started, that we calling it as where it open a space, that is called as micropyle. Inside the integument, parenchyma, diploid parenchyma tissues, that is known as nucellus. Diploid tissues, diploid cells, inside the integument, inside ovule, is called as nucellus. At the center of the nucellus, the female gametangium. The female gametangium is present. That we call it as embryo sac. Embryo sac types are there that I already told you: polygonum type, enothera type, and based upon the spores formation, either mono, bisporic, tetrasporic. How many number of the spores are participating in the formation of seeds, embryo formation? Okay, that also we will discuss. Okay, and then here you can see already usually what are the embryo sac type? I already told you, polygonum type of the embryo sac. Polygonum type. In that polygonum type, what happening? What is there? There is a 7 cells: egg operators, synergids, antipodal cells, 3 antipodal cells, 3 egg operator cells, middle to central nucleus or polar nucleus. 3 plus 2 plus 3. 3 plus 2 plus 3. How many? 8 nuclear embryo sac will be formed. That is a common polygonum type. That is first discovered by Strasburger in the Polygonum divaricatum. Polygonum divaricatum. That's why the type name is called a polygonum type. It is a common type. 70 to 80% of the embryo sac will be like this. 7 cells, 8 nucleus, 3 plus 3, and middle 1 cells. But in the middle one, 2 nucleus are there. 7 cells, 8 nucleus, embryo sac is the common type. Common type of the embryo sac is polygonum type. What type it is? Polygonum type of embryo sac. Okay, and then we can see the megasporogenesis. How the megaspore is generating. You see, single hypodermal initial. Single hypodermal initial, That means primary archisporial cell. Primary archisporial cells. As like a microsporangium. Primary microspore mother cell. Here the archisporial cell differentiate in the apex of the nucellus. Primary archisporial cell divided in pericliner. The outer one is called outer one is the form the cell wall, and inner one is form the mega sporogenous cell. Primary sporogenous cell. You see here how it is. The primary sporogenous cell. Yeah, is it able to see? You can see here clearly, very clearly, the structure of it. How it is modifying. How it is modifying, you can see here very clearly. You can see very clearly the structure. Archisporial mother cell. Here it is taking the division and it forming the what it is megaspore mother cell. The megaspore mother cell is taking the division. It is forming the. It's taking the what type of the division it is? Meiosis reduction division. So that it formed the 4 spore. It formed the 4 spores, divide into 4. Okay. After that, from the single megaspore mother cell diploid, single megaspore mother cell, it taking the reduction division, forming the 4 megaspores. After that, chelagellend 3 will be degenerated. Micropylar and 1 megaspore will be remain. The micropylar and 1 cell, again it take the first mitotic division. It formed 2, and the next mitotic division, and then it formed the 4 cells. And then later it again take the another mitotic division, not the meiotic division. Usually, there are so many competitive exams they ask. In the ovule formation, seed formation, how many number of meiosis and mitotic division will take place? Usually, the common type of the embryo sac, there is a 1 meiosis and 3 mitotic divisions will take place. Commonly, in almost all the type of embryo sac, that means megasporangium, female gametangium, only 1 meiotic division will take place, and remaining whatever either 2 or 3 mitotic divisions will take place. Okay, so that we can easily remember, recognize how many nucleus are present, how many cells are present in the embryo sac. The embryo sac that we can see easily, we can recognize. You see, first meiosis, meiotic division, dividing into 4. Of the 4, 3 are degenerated, only 1 will be remain. And the first mitotic division divided into 2, second mitotic division divided into 4, third mitotic division divided into 8. 4 micropylar and 4 chelagellend. Okay. And here the dislocation of these megaspores will take place. What happened here is, of the 4 from the micropylar and 3 will come together and it form the egg operators. And the 1 nucleus from the micropylar and will come nearer to the middle. And from the chelagellend 4 nucleus also 1 nucleus will come to the middle and fuse with this, so that diploid nucleus, secondary polar nucleus is formed at the middle. Secondary nucleus or polar nucleus will be formed with the two nucleus. The remaining three of the chelagellin will form the antipodal cells. It's like a vegetative cell. There is no use of these antipodal cells in the reproduction. Okay, in the reproduction, these two cells, egg operators and the polar nucleus or secondary nucleus only will take part in the fertilization. Okay, that only in the angiosperm. In the gymnosperm, only one fusion of the male and with the female. That is what the syngamy only will take place. Single fertilization. Whereas in the angiosperm, double fertilization. Here you can see the megasporogenesis. Based upon that, types of the megaspore are there. Types of the megaspore, that means types of the ovule are there. And here you can see types of the ovule, that is very, very important. Types of the ovule. These are the 6 types of the ovules. You see, orthotropous, orthotropous ovules. Orthotropous ovules. The first one is anatropous, orthotropous, compylotropous, succinotropous, hemianatropous, amphiotropous. These are the types of the embryo sac. You can see the very primitive type of the embryo sac. Very primitive type of the embryo sac is present in gymnosperms and some of the ranunculacea and so on. Okay, you can see here, orthotropous, it is straight line, funicle and microphyllarian. These all are in embryo sac, 3 in straight line. That is called as orthotropous ovule. Okay, and the next is anatropous ovule. Here ovule just curved 90 degrees. Curved 90 degrees. That is calling it as anatropous ovule. Here microphyllarian, funicle, hilum, embryo sac are very near, adjacent. This type of the ovule is called as anatropous ovule. Okay, and then hemianatropous. If it is turned 180 degrees, embryo sac, if it is turned 180 degrees from perpendicular to horizontal, that is called as hemianatropous ovules. Okay, and the next is amphitropous ovule. This is very, very important. Amphitropous ovule. Before that, compylotropous ovules. Here you can see embryo sac are curved, U shape. Embryo sac is curved, U shape, due to curving up to 180 degrees. Ovule is curved, but embryo sac is straight. Ovule is curved 180 degrees and curved, but embryo sac is in straight. That is compylotropous ovule. These all are present in pulses. And you can see the amphitropous ovule here in this ovule as well as embryo sac. Both are curved into U shape or shoe shape. Curved embryo is present in so many competitive examples. Curved embryo. Embryo curve into U shape. Along with the ovule, embryo also curve. That is there in the Amphitrachus ovule. Here, horseshoe-shaped embryos. What type of the embryo? Horseshoe, Kurrapu nada. Horseshoe type of the ovules are present. Whereas in the Sarcinotropous, extra round, extra sheath of the funicle. Funicle surrounds the ovule, which cover the ovule. That is Sarcinotropous type of the ovule. These are there in the open CR, which covering, which protecting the embryo ovule. You see, I already told you, orthotropous ovules, how it is, orthotropous ovules. These are our state, micropyle, embryo sac, funicle, three are in straight line. This is the primitive type of the embryo sac. That means primitive type of the ovule. These are there in the you can see these are there in Geridaceae and Piperaceae, Urticaceae, Polygonaceae, especially Piperaceae and Polygonaceae. These type of the ovules are present. The Polygonaceae and Piperaceae, these type of the primitive type of the ovule, that is state, micropyle, embryo sac, and conical, three are in straight line. That is happen. That is there in the primitive, that especially in the Piperaceae and Polygonaceae family. Okay, and then the next is Anatropous ovule. Here, what happened in the Anatropous ovule? Ovule is curved 90 degrees. So that what happened? Yeah, totally it curved into 180 degrees. State is turned into down. Upper part will going to down, down part will go to up. 180 degrees to 180 degrees, half of the circle. 180 degrees. Okay. And then what happened? Here the tunicle, hilum, micropylar and even embryo sac are very nearer to each other. Very nearer to each other. Micropyle and funicle are both side by side. Funicle, because funicle curve. You know, this is the common type of the ovule. Competitive exam they ask, which among is the common type of the ovule? Orthotropous, anatropous, comparatorous, sarcinotropous, semi-anatropous. What it is? Anatropous ovules are common type of the ovules, which is the common most type in almost majority of the plant. That's why anatropous ovules are the common type of the ovules. Okay. These all are there in the common in monocots, Archiclamidae, Sympetale, in all, most. Okay. And the next is hemianatropous. Here, what happened? The ovule body bend into 90 degrees, not the 180 degrees, 90 degrees, 90 degrees, perpendicular. Funicle is straight perpendicular to the ovule body, 90 degrees to the funicle. Ovule is 90 degrees toward the funicle. That type of the ovule, we call it as hemianatropous ovule. Here you can see the funicle and micropylar and just away than the anatropous ovule. Then the anatropous ovules. Okay, these are there in the ranunculus, Nothoscodium, and Thalbagia. In these three, especially in the ranunculus, note Nothoscodium, ranunculus, Thalbagia. In this hemi anatropous type of the ovules are present. Okay, and you can see the compilotropous ovules. Here, what happened? Ovule will be look like a horseshoe. Ovule will be look like a horseshoe. Why? Here, the compilotropous ovule body is turning up to 120 degrees. Up to 120 degrees. So that here also the micropylaran hilum funicle is adjacent, side by side. Okay. The compilotropous type of the ovules are present usually in the Leguminaceae, Fabaceae, Zabnacea, Mimosaceae, Leguminaceae family. We can see these curved embryos slightly curved. C is slightly curved. Okay. Okay. And then you can see the next. What it is? Amphitropous, very, very important. Amphitropous ovules. Amphiporous, amphitropous ovules. You see what appear here? Embryo sac and ovule. Embryo sac and ovule both are curved. Ovule and embryo sac. Ovule and embryo sac both are there. Both are curved. It looks like a horseshoe shape. Horseshoe shape. These type of the ovules are called as what type it is? Amphitropous type of the ovules. Okay, and these are there especially in Alismataceae and Butamaceae. Amphitropous type of the ovules are present in Alismataceae and Butamaceae. That is very important. Amphitropous ovules, that means curved embryo and ovule, are present in Alismataceae and Butamaceae and Centrospermaceae. So many exams they asked. Amphitropous ovules. Embryo is in curved shape in amphitropous, compilotropous, arsinotrophous, anatropous, atropous. In which one? Amphitropous curve is bended into U shape. Whereas in the compilotropous, only ovules are bended U shape, but embryo sac is in state. That is the difference between these two. You should remember that all. Okay. And then sarcinotropous. That means extra layer. Funicle is extended their growth and it covering the ovule. Again, it touches at the same place as the micropylaran, but it round around, it takes long growth in the of the funicle. Okay, that means here sarcinotropous ovule is curved 360 degrees, all 360 degrees. Again, it comes in the same position. Whereas in the hemianatropous, amphitropous, bend at 180 degrees. Whereas in the anatropos, it bends into 90 degrees. It's very, very important. Ovule turn into 90 degrees. Which ovule is turning into 90 degrees? Anatropos. Which ovule is turning into 180 degrees? Amphitropos, hemianatropos. In which the embryo is curved in horseshoe shape. Amphitropos. Alismataceae, Bitamaceae. And the Sarcinotropos ovule, where the funicle is surrounds and covers the ovule, and it turns into 360 degrees. 360 degrees. These are the types of the ovules which are there, and so more functions of the ovules are there. No main function is for the fertilization, and in that nucellus type, nucellus, that is the inner part. There is the inner part in the ovule, in between the integument and the embryo sac. In between the integument and the embryo sac, the diploid nucleus came from the megaspore mother cells that belongs to sporogenous cells that are called as nucellus. Nucellus, it is a nutritious layer. It's a diploid. This will supply the nutrition to the developing embryo, toward the developing embryo sac. That is the nucellus. We also called it as ovulus tapetum. Ovulus tapetum. It is also a nutritious layer. The endothelium, that also called as ovulus tapetum. This endothelium is forming from the nucellar tissues. It supplies the nutrition toward the developing, that means the maturing embryo sac, to the developing and maturing embryo sac. Okay. Developing or maturing embryo sac. Okay. That is the nucellus, the nutritive layer, nutritive tissue, which are present in the ovule, That is, you can see the nucellar structure, how it is, how the nucellar structure, how it is developing, how it is getting mature. The nucellus, it is also essay question. Nucellus. You can see here the nucellus structure, how the nucellus is forming, developing from the archisporial cells. You see, perithecial cells and megaspore cells. You see, the perithecial cells which are being evaluating, and it will become the nucellus cells. You see, which is covering the embryo sac, is covering the megasporophyte, which is covering the nutritious layer, which supply the nutrition to the embryo sac, is the diploid nucleus that we're calling it as nucellus. Nucellus, again, two types are there. It is the largest part of the ovule. The competitive exam also it asks. Largest part of the ovule is called as nucellus. Okay. Where there is a persistent part, remaining part, even after maturation of the embryo sac, the remaining nucellar part, we call it as perispore. Remaining nucellar part, we call it as perisperm. Usually, the perisperm is present in Cyperaceae, Polygonaceae, and the black pepper and beetroot. Beetroot. In there we can see the perisperm, and the remaining nucellus, even after maturation of the embryo sac, is called a perisperm. You see, depending upon the development of the nucellus, the two types of the ovules are there. That is also very important. Based upon the nucellus, grassy nucellate and tenunucellate. You see, grassy nucellate. What happens here in the grassy nucellate? Nucellus, here you see more number of the parietal cells or periclinal divisions will take place. Several members, you see, the more number of the parietal division, that means more number of the nucellar tissues. If it is a single layer parietal nucellar tissues are there, that we call it tenunucellate. Tenunucellate type of the nucellus is present in the Rubiaceae, Asteraceae, Orchidaceae. You know that because of the grassy nucellate ovules, Cucurbidaceae family is kept in polypetalae. Though fused petals are there, because of the ovule structure, nucellar structure, the Cucurbidaceae family kept in the polypetalae as per Bentham-Walker classification. That is, grassy nucellate is the primitive, tenunucellate is the advanced one. Okay. And another one is pseudo-grassy-nucellate. No partial cell is present. Only epidermis will be divided into multi-layer. Okay. Here the formation of grassy-nucellate and tenunucellate of this. Here you can see the integument we already studied. Integument, based upon the tegument, already told you three types: ategmic, unitegmic, and bitegmic. Ategmic integument, if it is only one layer of the integument, if there is no integument, there is no layer, there is no tegument layer, that we call it ategmic. The ategmic is there in, you see, Santalum and Loranthus. You see, in this, the ategmic, no tegma option. And unitegmic, only one layer, single layer tegma. If the single layer tegma is present in ovule, that is called as unitegmic. Okay. Integumentary tapetum. You can see these are there in Compositae family, Asteraceae family. And bitegmic. Usually most of the ovules which are in the two tegmas, inner and outer. Bitegmic is common, primitive. These are occurs in the monocots and the primitive dicots. It is present. Ditegmic. Okay, based upon the tegma, based upon the layer, ategmic, unitegmic, and bitegmic. Here you can see only one layer, unitegmic. Here you can see the multiple layers, two layers, bitegmic. Okay, that is the tegma integuments type. And you can see the extra structures of the ovules. That means extra layer other than the integument, extra layers also present. You see extra integumentary layer, we call it as eril. Eril that we eat in the Pithecanthrobium dulcis, Simachinta, Gubaka antama Simachinta. The Pithecanthrobium dulcis, round, red color. That it seed is covered with the eril. That eril we eat. Seed covered with the eril, extra integumentary part. That is called as eril. You know, even in the Georgica Japatri, Japatri, what we take it out from the Georgica Meristica fragrans. The Japatri we use for the fragrance in the biryani, for the digestion also, in the biryani also. That also eril, extra integumentary part is called as eril. And the other one is caruncle. There is extra structure, mucilaginous structure at the micropylar end. At the micropylar end, mucilaginous extra seed at the micropylar end is called as caruncle. Not the bus uncle, car uncle, cycle uncle, scooter uncle. Okay, caruncle. Caruncle means it is a extra layer, extra mucilaginous seed, mucilaginous sacs are there at the micropylar end. That is called as caruncle. That is there in the resinous communis. You know the resinous communis castor, amudam. In the castor amudam we can see. And also another extra structure is comos. Another extra structure is comos. Comos means you can see hairy-like structure. Here you can see the hairy structure, especially in the Asclepidaceae. Gilled, we can see in the gilled. Extra comos, airy, feathery structures. With that, these are useful to disperse the seeds. Aerial, caruncle, and comos. These three are extra integumentary parts. This extra integumentary part is calling it as is useful in the dispersal of the seed. These all are aerial, caruncle, comos. These three are useful in the dispersion of the seeds. Okay, and you also can see endothelium or integumentary tapetum. That I told you. You see, here you can see in this, whatever the integumentary layer, that integumentary layer, sometimes it is what happening? It is forming as a nutrient layer, as like a tapetum of the microsporangium. It also supply the food material, transportation of the food material from the integument toward the embryo sac. Integument toward the embryo sac. It acting the nutritive layer. It supply the nutrition to the embryo sac. It also give the protection. It also give the protection. That is the also looking like a seed coat. It supply the extra nutrition toward the embryo sac. And then another extra structures are hypostage and epistage. Hypostage and epistage. Here you can see the hypostage, which are there in Amaryllidaceae, Liliaceae, Gingiveraceae. You see where there is a basal structure which are there just below the embryo sac. Just below the embryo sac, which prevent the penetration of the embryo sac toward the below It gives the stable position to the embryo sac. That is hypostage. Hypostage. And then next is epistage. It is there just above the embryo sac. Just above the embryo sac, which protect, which give the specific position and which stop the entering of the extra pollens that is there in the embryo sac, in the ovule, at the above the microfilar end to prevent the extra penetration of the pollens. Okay. You know, operculum structures. These are sponge-like structures outside the seed coat. The sponge-like structure which is formed outside the seed coat that are called as operculum. And arboretum. Arboretum is very, very important. Arboretum also. This is also there at the micropilar end. This is also there at the microfilar end. It gives support, strengthen. You see the arboretors, obturators. These are the obturators, finger-like projections at the micropilar end. Here at the micropilar end, you can see the finger-like projections. It rays from the placenta, grow along with the ovule, and it finger-like projections. Okay, but it lacks vascular tissues. It look like a glandular cells, and it give the support, and it also useful to reach the male gamete toward the ovule, ovary. They show the way to the pollen male gamete. That is the characteristics of the obturator. Okay. Integument, nucellus, and extra integumentary parts. What are those? Aerial, caruncle, comos. Epistage, hypostage. Epistage, hypostage. Okay, operculum, obturators. Okay, and integumentary tapetum, also called as endothelium. Whereas in the microsporangium, we studied endothecium. Endothecium. Okay, outer layer of the microsporangium. Here, the integumentary, some of the integumentary part is forming as a nutritious layer that is called as endothelium or ovulary tapetum. Ovulary tapetum or endothelium. That is there in the ovule. Endothecium is present in the anther. You don't confuse between the endothecium and endothelium. Endothecium is present. Endothecium is present in the anther, outer layer, middle layer, tapetum, endothecium. But whereas the endothelium is the integumentary part, the nutritious integumentary part, that is called as endothelium or ovulary tapetum. This is there in the ovule, this is there in the anther. So don't confuse. Okay. And then the main important part in the ovule is embryo sac. That means female gametophyte. Embryo sac or female gametophyte. The embryo sac or female gametophyte, which is present inside the ovule at the middle, integuments, nucellus, and embryo sac. Embryo sac, we have studied the types and embryo sac structure. Embryo sac is 7-cell 8 nuclear structure. 7-celled 8 nuclear structure embryo sac. That is that. Okay. And then we can see here, already told you, egg cell operators, antipodal cells, and polar nucleus. The common type of the embryo sac is polygonum type of the embryo sac. That I told you already. Okay. And then shall we go for the megasporogenesis? The megasporogenesis, which is there, which is happen in the ovule, how the megasporangium is forming. The megasporangium formation, that also we will take, that also the essay question. The megasporogenesis. Megasporogenesis. Okay, megasporogenesis, that is happening in the ovular structure. They based upon that, these are the types of the megasporogenesis are there that we will discuss. Here you can see the megasporogenesis, how the embryo sac type, embryo sac types and megaspore type, megaspore types or embryo sac types, that we will discuss here in the ovular structure, megasporogenesis. Here you can see the megasporogenesis, how the megaspores are forming. The megasporangium. This megasporangium is forming, already just discussed lightly, is from the archisporial mother cell. From the archisporial mother cells, megaspores are forming, the megasporangium, the parietal cells and the archisporial cells. The parietal cells, which are, if it is forming the single layer of the parietal cell, that is 10 nucleus layer. If it having the multi-layer of the nucleus, that we call it cross-nucleus, that we are already studied. Megaspore mother cell. The megaspore mother cell, it take the division, the megaspore mother cell, based upon that, it having the multiple types of the embryo sac, megasporogenesis are there. You can see in the megasporogenesis, during the process, how many number of the spores are participating in the formation of embryo. Based upon that, there is a 3 types are there. That is monosporic, bisporic, and tetrasporic. Monosporic, bisporic, and tetrasporic type. What are those 3 types? Monosporic, bisporic, and tetrasporic type. 3 types. If after the meiosis, if only the 1 megaspore is taking place in the formation of embryo sac, that is monosporic. After the meiosis of the 4, if 2 megaspores is taking place in the formation of embryo sac, that is bisporic. After the fertilization, if the 4 spores are taking place part in the development of embryo sac, that is tetrasporic. These are the 3 types of the embryo sac. These are the 3 types of megasporogenesis that is happening in the angiosperms. The angiosperms. Usually, in the megasporogenesis, what happening here is, what happened here you can see in the first one, monosporic type. That means in the monosporic type, what happened? In the reduction division, megaspore mother cell, megaspore mother cell, megaspore mother cell, this one, MMC, is taking the reduction division, that means meiosis. and it forming the 4 nuclei. Of the 4 nuclei, 3 will be degenerating at the chalazalan, and microphylla will be remain that single nuclei of megaspore mother cell is taking further division into 2, and next mitotic division 2 into 4, and the third mitotic division 4 into 8. Of the 8, 4 is at microphylla, 4 is at chalazalan. The microphylla 3 is forming the egg operators. Microphylla 3 is forming the egg operators. That means egg cell surrounding with the 2 subsidiary cells. And the chalazalan 3 nucleus are forming antipodal cells, 3 antipodal cells, vegetative cells, and the 1 nucleus from the microphylla, 1 nucleus from the chalazalan coming together, and it forming the dichoretic 2 nucleus polar nucleus, polar cell is forming. So here you see the 3, 3, 1, 7 cells, 3, 3, 6 plus of the middle 2 nucleus, 8 nucleus, 7 cells, 8 nuclear embryo sac is forming in the monosporic type. It is the primitive type. Common type of embryo sac is called a polygonum type. That also we have discussed. Okay, and then in the monosporic again 2 types are there. That is polygonum type and enothera type. Polygonum type and enothera type. Here you can see in the polygonum type, chalazalan megaspore is remain and is taking 1, 2, 3 mitotic divisions and is forming 8 nuclear embryo sac. Whereas in the enothera type, only 4 nucleus is present, not the 8 nucleus that are, you see, after the division, microphylla 1 will be remain, that is taking the only first mitotic division and second mitotic division. With that, it is forming only 4 nucleus. Of the 4 nucleus, 3 is forming egg operators and 1 will be at the polar nucleus. There is no antipodal cells. There is no third mitotic division. So here the polygonum type it is 8 nucleus, enothera type is the 4 nuclear states. And then the next is bisporic type. What is that? Bisporic. That means after the meiosis, 2 spores, 2 megaspores are taking part in the formation of embryo sac. In the formation of embryo sac. After the reduction division of the 4, 2 is degenerating, 2 will be remain. The remaining 2 is taking